Axel.
Do I just start? Do you want to do an intro? How's this work these days?
Yeah, you can just
Okay, cool. Hi everyone. Hi everyone on Zoom. If you guys don't know me, I'm Axel. I am a postdoc, although officially I'm assistant specialist in the knee lab in the Department of Maths, and I guess what was formerly CMCF. Today I'm going to be talking about a paper that had basically taken up the better half of my postdoc and was recently just out in Nature Methods, and Nelly kindly invited me to talk about, and kind of also sums up a lot of what my lab, the knee lab, tries to work on in a sense. So yeah. Sorry if I stop. Whoops. Okay. So just as a bit of biology from a non-biological expert, all of this work and in general the work I'm trying to do in the future is based on a simple premise that no cell's an island. The data we get these days, you usually try and analyze what's going on in a single cell or a group of cells, but we all know in reality, cells are continually sending and receiving messages from each other. And there are a number of ways cells transmit information to each other. The main form I'm interested in is cell-cell communication through what is effectively ligand-receptor binding. So cells secrete molecules called ligands. They primarily diffuse through space. They can also be transmitted across cell junctions. They bind to corresponding receptors. And usually what will happen is if there's enough signals being sent, they will trigger some sort of downstream reaction. And so the idea is like when you have these ligand receptor binding processes, they effectively generate what we in the lab call flows of intercellular information. So you have a single cell, say a ligand, the blue thing binds to the orange receptor and triggers some sort of downstream effect. So certain genes get transcribed, and it also may even cause an additional flow of other ligands, other molecules. The idea being here is that cells are receiving messages and it might cause them to send other messages. And so the key question I spent a lot of time with Ching trying to figure out is how do you infer what we're calling these intercellular flows? How do you infer these dependencies generated by the most common form of cell-cell communication, ligand-receptor interactions? And the key caveat to this question is how do you infer intercellular information from data where it is not quite obvious how do you kind of measure or infer this? Since this is GenPALS after all, this is really all about how do we infer these sort of intercellular flows from genomics or transcriptomics data. So I had this slide because I was presenting at a different event, and the people here who attend these talks are a little more in the know, but basically these days we can kind of use NGS technologies to do whole genome captures, either at the single cell or spatial resolution. Basically, at the end of the day, as a mathematician, all you get is a giant matrix. And the reason and what's nice about this is that you can effectively look at known ligand receptor interactions on the order of thousands, whereas previously it was, say, on the order of tens and maybe hundreds if you were pushing it. And the key novelty here is that even though we're using transcriptional information, you can actually infer cell-cell communication based on ligand and receptor gene expression with surprising accuracy. There's a lot of very nice work done by people at UCI and the Skin Neuro Labs that have kind of gone and verified that these methods work. And UCI, as you guys might know, is also pretty good at developing methods that infer cell-cell communication. So yeah, we started my lab, the knee lab, started looking at cell-cell communication in like 2020. I believe cell chat, like when I first joined, Switching was already developing cell chat and it was in preprint mode. During the pandemic, we got asked to write a review about cell-cell communication at the time when there weren't many methods. In 2020, at the end of 2020, we counted about 19 methods. Fast forward about 3 years later, that number is about 105, and I think it's still growing. The point of this slide really is that it's still an active field of development, even though a lot of the state consensus methods were developed in like 2020, 5 years ago now, which is quite a while. Yeah, and this Armingol review by Nathan Lewis is probably the most up-to-date comprehensive method or review. Yeah.
Here, with that, I want you to take a picture of it so I know what paper it should go.
Yeah, I would love to if I could figure out. Yeah. And I guess I'm pointing it out as also our lab had got asked to review that review, so that was fun. And again, despite all these methods, I should say, like, there's still so much we don't know about cell-cell communication. Last week, there was a talk about metabolomics, which is a huge part of cell-cell communication, and which we all know we know very little about in the context of single-cell spatial technologies. There's also people in my lab working on trying to infer like metabolic communication. So there's lots to do still, despite all of this. All right. So in case people don't know much about cell-cell communication methods, these are the sort of two prototypical methods that we use that were coincidentally developed in the knee lab. The first is cell chat, which I guess a lot of people have used or seen in some form. Basically, you take single cell data, which is a giant matrix, you have some predefined annotations, usually cell types or cell states, and you can infer effectively these networks generated by ligand-receptor interactions. There's a certain formula used to calculate the chance of cell-cell communication occurring, or the interaction strengths. And the key novelty with this method, it wasn't so much the inference of cell-cell communication, it was more the different network analysis tools. And it was the first method to really kind of develop a proper suite of visualization for cell-cell communication. And as a result of this method, there's other versions like neuron chat, because with neurons, they communicate in a slightly different way. By small molecules. You guys will know much more about it in the room. There's MetaChat that we're working on, and there's also been cell chat developed for other model organisms. Originally, this was developed for hare and mouse and human. There's also ones developed for chicken, zebrafish, I think even axolotl now. So yeah, the idea is you put in the matrix out, you get a bunch of networks that you then need to decipher and kind of do some meaningful analysis. That works. So in terms of space, it's a little bit different. Spatial inference methods have, I would say, a far fewer in number than non-spatial methods. Mainly the reason being is that with most spatial technologies, you lose the single cell resolution, although with technologies like stereoseq, slide tags, it's getting a little better. But here the idea is what you lose in, say, single cell information, you gain in spatial resolution. So the key caveat with single cell methods is you don't actually know if the communication makes sense, because cells can only communicate with those that are nearby. With spatial data, you have the spatial information, and so you can actually do something nice, which is if you have, say, distributions of ligand receptor genes, you can actually predict where the interaction was both sent and received using like optimal transport methods. The idea being here is like you can also account for the fact that multiple ligands can bind to that same receptor, or multiple receptors can receive the same ligand. So this is a method I think Jishen, former colleague, came up with in 2021. And also people at UCI helped validate. So it was a very nice piece of work in the end. And so the idea, so there's a lot of these are sort of the two types of methods you get for inferring cell-cell communication. A key caveat of them is that we tend to infer ligand-receptor interactions in a one-by-one basis, and that we tend to look at, say, Wnt signaling as being independent of, say, BMP signaling. And the reality is that we know in general, in development, in disease, there's all sorts of tightly like regulated dependencies between these events. And so the question we had at the end was, how do we, given all these ligand receptor interactions we can infer, how do we infer which of these actually depend on each other? And can we do this directly from the data without any sort of pre-assumed knowledge? And so, actually, I'll go back, oops. And so the question we initially had was like, okay, if you know, for example, you have fibroblasts and macrophages, and fibroblasts signal to macrophages via Wnt signaling, and then you see macrophages signal via, say, BMP, how do you infer the effect of macrophages receiving Wnt signaling, and how do you infer whether that drives the outflow of BMP signaling? That in itself is kind of a hard question, because you have to understand you're asking if what another cell is doing is affecting the cell in question. The way we kind of got around this is by reducing the specificity of that question, but making it kind of about the same cell. So the idea is rather than asking what is the effect of fibroblast sending a signal, you're asking what is the effect of receiving a different signal. So if I receive Wnt signaling, does that then cause me or drive me, result in me sending BMP signaling? So you're kind of flipping the question, and you're making it about, say, one cell rather than, say, two different types of cells. if that makes sense. So yeah, go
I'm
on.
so confused now.
Go for it.
Can you rephrase that again?
Yeah,
So how do you reduce? I completely understand what you are just talking about. How do you reduce from that complex problem again to a more simple question that you are asking? That's the gap that I was
Oh,
really
no worries.
unclear how you actually do the jump there.
This is probably the best slide to explain it. So here you have three groups of cells, A, B, and C, and let's say A signaling to B, the orange cells causes B, the group of cells B to signal to C, the green cells. So the question to ask is like, did A communicating with B cause B to communicate with C? That was the initial question we had.
That's so hard to figure out.
Yes, that is a really hard question. So rather we asked a simple question, which was like, does B receiving that first signal cause it to send the second signal? So the key thing is you're only talking about measurements within one cell group, B,
Okay,
the middle group.
okay,
And so rather than thinking about like, oh, so I lose the fact that like I can't say whether the signal came from A or not, but I'm thinking about what other things that come in versus what goes out, and it's all
Okay.
within the same cell.
okay.
The signal that is coming from A, which we don't know because we're removing, how do you know it's like, it must be like an accumulated effect of multiple senders
Yes.
versus like an isolated A.
Okay, yeah.
That is an excellent question, and the truth is we don't know. Basically, all we know is like receiving one type of signal. Yeah.
Which is still very informal, but like just knowing like this is the signal that this cell is getting and this is how it's responding to it.
Yeah.
It's still very, it's hard to quantify and find out the sources of the.
Yes, this is where as it currently stands, you basically have to backtrack through your cell communication analysis and be like, what were the original sources, if I were to answer on the spot. But you're right in that kind of disentangling the specificity of those signals is still an open question. Yeah.
So what I think maybe you're pointing out here is instead of saying this direct interact with leading to that result,
Yes.
you are focusing only on this.
Yep.
So in cell chat or some other tools like cell chat only focus on this part.
Yep.
Or this part.
Yep.
You are actually focusing on possibly this method or this ligand receptor interactions reduce something for this and leading to a result here. So which
Yes.
is only these cells.
Exactly.
So you are switching up. So the major question you're asking is from A, B, C. But you're actually only focusing on this one, which narrows down to the signal only on B.
Yes.
Okay.
So this is the same reason that why like gene regulatory network inference works a lot better for single cell than say cell communication. So gene regulatory networks, you're still trying to say something causal about what's going on, gene A affecting gene B and gene C. The reason you can do that is they're all measurements within the same cell.
Yes.
So whereas the key challenge with cell communication is that it involves multiple cells. So we like basically, yeah, the key insight was like you reduce the complexity of the question so that you're still thinking about one cell or like the same cell, but it's still a question of like signals that are received somewhere else.
Yeah, even for signals that coming from other cells for gene regulation network, it has to get into that, let's say cell B in order to functional in the cell B.
Mm-hmm.
Yeah.
Exactly. Yeah. Oh, sorry, I went ahead, so now I got this run.
Here, because this is a complete, you think about the question in a different angle to reduce the complexity for this.
Yes. Yeah, there was an ideal question we would have loved to answer and we realized it was basically impossible. So, yeah, so test the effect of received signals on sent signals within the same cell. So yeah, it's a slightly different question, but yeah. All right, so this led to us developing this method called FlowSig, which infers what we're calling communication-driven intercellular flows. The idea is you take gene expression data, the key is both raw and normalized. For its spatial data, you can just take like one slice or multiple slices. If it's single cell non-spatial, you need a control and perturb comparison. So like wild type and stimulated for reasons I don't have time to get into, but it works better with non-spatial data. you take, when we developed it, I was like, well, I'm not going to reinvent the wheel. We'll take cell-cell communication output from cell chat or comma. In the future, I would probably refine this method so that you don't need to have previously inferred like cell chat. But basically at the time, gene expression, some cell-cell communication inference, a list of ligand-receptor interactions basically, and you spit out this network, which effectively have three types of nodes. At the top are called signal inflows. Those measure the different types of signals that you receive. So like did you receive like Wnt BMP, sonic hedgehog signaling? And then when you receive those signals, you process them at the intracellular level, so via some gene regulatory networks. To kind of speed up computational inference, what we did was rather than talk about individual, say, transcription factors, which are also very hard to get good estimates from transcriptomics data, we thought about it rather as sort of like intracellular modules or programs. So you can think of it as effectively like matrix factorization, if you guys are familiar. You have these modules that are made up of multiple genes that are based on co-expression. And so the idea is these modules, like these co-expressed genes, contain some information about intracellular processing. It was just a way of kind of coarse-graining the approach. And then finally, after the cell has received the signal, processed it will spit it out in terms of like in what we're calling signal outflow. So that's what's the easiest to measure. It's just direct gene expression or signal ligand gene expression. Go for it.
Um, Under what circumstances would you suggest to use raw gene expression data? Because normally it's recommended to at least do a library size
Um,
sure the effects are.
So the actual inference is done on the library size normalized and variance stabilized data, so standard normalized and log transform. The raw is needed because all matrix factorization methods require raw UMI counts. That's just the way they work, because usually the models are based on, they usually assume a Poisson distribution. So for the methods we use, it requires raw data. You're right in that, for example, library size normalization matters most with things like differential expression. My opinion, and it is only an opinion, is that like we're all the field is slowly moving away from doing things like log transforming so that we can do things like PCA and more about developing methods that actually describe the data, which is the fact that like single cell data is discrete at molecular counts. So rather than like 1, 1.2, 1.3, it's like 1, 2, 3. So it isn't an either or, you actually need both for this method. So yeah, as it turns out, the methods that generate the intracellular modules all require actual raw untransformed counts. They do some pre-processing, and there is some normalization within those methods, but yeah, that's the main reason.
Can I ask one more question here?
Yep.
Maybe you're welcome to talk about that later. So, I was actually checking the tutorial on the README right now, because I really want to use this.
Oh, you do? There you go.
Because the question I can just tell you right now, since we're on the same lab, basically. So we found out a very interesting things in the neurodegeneration. We found out one transcription factor that is technically downregulated in oligodendrocyte. And if we actually upregulate the oligodendrocyte transcription factors, oligodendrocyte actually talks more with astrocytes to reduce or to reduce some activity for microglia, which is a very bizarre thing for me in the beginning. Is how oligodendrocyte actually talks to cell A, talks to cell B, which is astrocytes, and then signal microglia in a way. So that's something I think is very suitable for using your package right now. So that gave me idea is not only in the beginning I was just going to run 2 times of this like cell chat, right? Oligodendrocyte to astrocytes, astrocytes to microglia, but we still don't know what is the the signals that flows from oligodendrocytes to microglia. So if we can use this, and do we need to run cell chat, communicate cell chat first, and then insert, because your tutorial is actually in basically ScanPy.
Yep.
Cell chat is in R in Seurat. So unless I don't know if there's a Python version of cell chat. So that means, do I have to run cell chat first and fit into your network?
Currently, yes. In the future, no.
Okay.
My goal is that no, it
So
will not be the
that's
case.
why you said, okay.
Yeah,
And following
but
up,
there is a Python implementation of cell chat, not
there
by
is.
our lab, but there are people who've come up with a Python
okay, is that
implementation.
consistently as well outperform the rest of the things from?
To let you guys in on a secret, there is no outperform. They're all different, and
They're all
that is a
different.
field of big area of concern for those who do cell communication
Okay, that
inference.
makes
Yeah.
sense.
So I guess also for you guys, your lab, you would use more neuron chat, right? Because they're a
We
long
actually
range
s-
communication.
Yeah, we actually stay with the cell chat now. The reason why is when neuron chat was first out, it was still in the alpha phase, and we got it and didn't work that well because there's bugging in there, so we didn't really switch completely. And
Mm,
after that, we didn't get other projects that actually use a lot of chatting communication necessarily, so we haven't been switching.
okay.
But probably we're going to. My biggest understanding between that two is the database get aggregated, or probably there's deeper algorithm that is get changed. But for my basic understanding is the database is a little bit different. Is that
Okay.
true? Or
For
for neuron chat and cell chat?
yes, they are different. So the
Yeah.
point is like neuron chat had different types of interactions that weren't just included in cell chat.
Yeah. So
Um,
the next thing for your question, for your is you're going to fit in this signal flow from cell chat. That's from cell chat, right?
Yeah,
And the internal modules, intercellular module is what you can calculate from your FlowSig.
yes.
Okay.
But if you're interested in specific transcription factor, you can also specify individual transcription factors, actually. Yeah.
Because for even for co-expression level, signal expression level for transcription factor is extremely low. So
Yep.
a lot of time they are not
It
actually
is, yeah,
included
dog
in-
shit for transcriptional data, transcriptomics data, yeah.
Yeah, so that's something I don't know how to deal with, to be honest.
Yeah, yeah, that's a really good question.
Because if we are using because you mentioned co-expression network, right? A lot of time those lowly expressed genes are in the gray module, which is they are not associated with anything else, which is very hard. Even for transcription regulatory network, sometimes you like I have to really loosen up for the TF expression level.
Yep.
To include them in the network. Otherwise, there's like sparsity just going to significantly reduce how many like transcription factor I can actually include in the network.
Yep.
So I don't know how to do this.
Honestly, okay. I don't know enough about your lab. Are you just looking at RNA-seq data?
I think we
Yeah.
can also talk about this after.
Um,
Yeah, please continue.
if okay, I'll give you a short answer is you sound like you're fucked.
Yes.
Long term, yeah, I'll think about it and then I'll get back to you at the end of this.
Yeah, thanks.
But you're not wrong. Everything you said about transcription factors is exactly how I feel about RNA-seq. Anyway, so yeah, you put in gene expression data, cell communication out, you get a network effectively of things that describe the inflows of signals and how it drives an outflow of other signals. Yeah, works for both non-spatial and spatial data. So there are three major steps in this. One is like, how do you define the different variables that you're going to do the inference on? Two is how do you actually learn the networks of intercellular flows? And three, what I've called pseudo validation, but it's really more just like a sanity check that things make sense. Okay, so in terms of step one, this is probably the bit where like relates to the receiving and sending signals. So there are three types of variables you construct from input. I think for this talk, I only use spatial data because it was for a spatial seminar anyway. So there are three types: signal inflow, the amount of signal received, intracellular module, which is like the enrichment for different regulatory programs, and three, signal outflow, which is the easiest, which is just how much of a gene that corresponds to a signal ligand are you expressing. Signal inflow, so spatial data, you kind of bypass the need for transcription factors because using Duchen's method, which is optimal transfer based, you can actually predict exactly where ligand receptor interactions were received. So if you know that, you could also like downweight this by transcription factors, but effectively this works just as well. So the idea here is that there's say this for the spatial data set, there were two inferred ligand receptor interactions involving Wnt5a. Wnt5a can bind to at least two. There are probably more receptors, frizzled 1, frizzled 2. The idea is Even though it's different receptors, effectively you're measuring the same thing. You're measuring how much of Win5a did you receive. So in this case, you just take the sum, you get that basically distribution on the right, which says how much Win5a flowed into the other regions. The reason this is just different from, say, expressing Win5a is the point that these signals diffuse in space. So you're accounting for the fact that sending a signal is not the same as receiving it. When you're sending, like Win5a is expressed, secreted, it diffuses and moves in space and then is received somewhere else based on, say, receptor expression, spatial constraints, and maybe even downstream activation. So effectively both non-spatial and spatial methods, components of FlowSig use a form of matrix factorization, which is effectively dimensionality reduction. The idea is you take this matrix, the original matrix, which is m by g, where g is on the order of 10,000, 20,000, and you're decomposing it into k modules, where k is on the order of like 10. And so you're hoping that the columns on the left describe spotwise or cellwise enrichment for each of these k modules, and those in the row matrices, the k by g matrix describes the enrichment for different genes, so the contribution of different genes. And what we're hoping for this is that each of these we're going to use the n by k matrix to describe enrichment for the different intracellular modules. And the idea is you're hoping, as it works with this case, that it encaptures some sort of meaningful, both spatial information in this case, but also meaningful cell type or cell state information. In this case, this is capturing important anatomical information in this embryo data set. Here we're using a very nice method called non-negative spatial factorization, come up by Will Towns, who's done a lot of nice work over the years for single cell and spatial work, and Barbara Engelhardt, if you guys have heard of her, also does a lot of very meaningful work in this area. And so, yeah, we're basically taking information like this, enrichment for different regions, cell types, genes, etc. And then the signal outflow is most straightforward. You're just taking different signal ligands or different genes and their spatial expression. In this case, it is like the normalized variant stabilized, so the continualized form of it all, rather than the raw expression counts. So you put all of these in together and you define, and you learn this network based on this very simple idea that there's this chain of dependency that occurs. So when signals flow in, they're processed at the intracellular level, and then they're sent out in the form of outflow. And so you receive a signal, process it, send it out. And so this sort of like chain of dependency underlies the entire network inference model and also the validation step afterwards. And so realizing this and that like all signals kind of have to obey this like pathway really simplifies the inference in the end. All right. And then two, this is probably the most technical bit, which I'm going to then, as a result, talk the least about. There are two steps to learn the networks. One is something called conditional dependence testing, which uses really correlation, but in a more, rather than talking about correlation between pairs of variables, A and B, you're talking about correlation between three variables. And so the idea is like, conditional dependence says if I have three variables, A, B, and C, if A and B could be dependent on each other, but if I know information about C, the third variable, suddenly A and B become independent. The simplest example I can think of is that the past and the future are independent given information about the present. So obviously present depends on the past, future depends on the present, therefore future depends on the past, but if you know information about the present, suddenly the past and the future are uncoupled. So the idea here is like this method effectively gives you where the edges should sit in your networks in an undirected sense, so you don't know where the directions of causality go. If you did just simple correlation testing, you would overestimate the number of edges in the network. And so conditional independence by testing three variables triplets, you basically, it gives you the right amount of edges to kind of cut a short story. And then second, which is probably the most contentious step, which is learning the directions of causality in this case. It is very well known in the field. If anyone's done anything on graphical causal models with Judea-Pearl's network stuff, which is that if you have a set of statistical relations implied by conditional independence, there can be like hundreds, if not thousands, of graphs that correspond to that relation, depending on the number of nodes. So it doesn't actually give you a unique answer, which is the fundamental problem. And so the big kind of topic of contention is like, how do you figure out the correct orientation? In this case, we're using an algorithm that was developed by Caroline Uhler's lab at MIT, which is based on something called a greedy sparse permutation algorithm. The idea is that in this case, to work out the correct graph often corresponds to working out the sparsest network, and also in a way where things are consistent with what's called the Markov equivalence clause. Basically, there's a certain set of directions you can imply from the data, and by searching in a certain space of networks that give basically the correct answer. I'm not going to go too much into it, but effectively, it works relatively well. And then the final bit, which is just making sure things make sense. So because we want to get some degree of confidence, you do a form of bootstrapping, you learn like an aggregate of networks and you average them. And then you remove edges in the network that don't appear that often, because basically we're saying they're low confidence. This is the other bit you can do. So, and then the final bit is we're reorienting edges according to their Markov equivalence clause. So something I didn't say is that the conditional independence relations, or the Markov equivalence class, gives you both directed and undirected edges. The undirected edges, if you orient them either way, it is the same statistical relations. So that's actually fundamentally a problem. How do you kind of know which way to orient those edges, the undirected ones? Luckily, we know which way we can based on the fact that signal inflows and signal outflows, there's a unidirectional chain of causality there. So basically, we just reorient edges so that they obey this simple relation that signal inflows lead to intracellular modules, which lead to signal outflows. And then, yeah. And so that effectively seems to work pretty well, this procedure, as ad hoc as it is. So yeah, this is just a recap. Gene expression plus cell-cell communication equals a complex network that actually works surprisingly well in practice, of describing the effects of receiving signals and on the effects of sending different signals. Okay, so this is a brief demonstration. Yeah. So I'm going to demonstrate this on a spatial data set generated by StereoSeq. I don't know if it's available at UCI. There was a Cell paper in 2022, which basically gave us spatial data at the single cell resolution and also at whole genome coverage. So it was a little better than what FlowSig can do. I think everyone in my lab has used this data set at least once for some sort of benchmarking. I think Max has sent postdoc Raul literally to Taiwan to get data like this. Yeah, it was developed in China. So currently, for all sorts of reasons, that technology can't be accessed in the US, but it can be accessed in Singapore and Australia, as I've learned. Anyway, it is a very nice study. They did embryogenesis from stage day 9.5 to like 16.5, and it's annotated. It has raw and transform count, works very well. So the idea is like we're taking this data, which is annotated, we generate spatial modules that describe roughly these different anatomical regions, but instead of this discrete representations I'm showing, we actually get continuous like relations or continuous variables that describe like the membership or enrichment for those different programs. So the ultimate like output you get from this FlowSig is actually usually quite complicated. It's a network of all different signal dependencies. You might have, which means you can, depending on the number of nodes you're looking at, like the number of variables, which ranges from like 10 to 100, the network can get pretty ugly to look at. What we found was more useful is if you focus on a particular variable of interest. So for us, because we're talking about embryogenesis and development, we thought of looking at Sonic Hedgehog, which turns up everywhere and is really important in morphogenesis. And we wanted to look at what FlowSig said about it. So we kind of broke it down to two parts. One was to look at what are the upstream regulators of Sonic Hedgehog. So what other signals, if you receive them, cause an outflow of Sonic Hedgehog, the spatial expression of which is shown there. In this case, FlowSig predicted that there were 8 different variables. I think ones that make sense, like BMP4, Win5a. Interestingly, FlowSig also infers that Sonic Hedgehog receiving Sonic Hedgehog drives an outflow of Sonic Hedgehog, which we'll also prove in a moment. Go for it.
My brain can wrap around the concept because embryogenesis is like a time-dependent thing, but the data that we have is a snapshot, right?
Yep.
So, but the method that you have like single being received and then extend, but that can kind of create a sense of time, maybe, like, I don't know, like how you go.
You have described literally what is like been in my research statements and is the fundamental flaw of all these models. It is a really good point, which is like we're trying to say something that is ultimately trying to make an inference about something that is ultimately dynamic from static data. And so that is like a major limitation. When you look at these networks, it almost implies like a spontaneous or like simultaneous like causality or dependency, when the reality is like you're right, like there's a time lag. Sonic Hedgehog secreted diffuses, that takes time, minutes, hours, it's received, and then it causes an outflow. That is like something that occurs over time. You're right in that like we lose all that information and we can't say anything meaningful about that at the moment. All we can really say is given what the snapshot gave us, which is usually some sort of slowly evolving timescale, this is what FlowSig and the conditional independence and graphical model inferred were like dependencies. So basically it's like it's kind of almost like an exploratory tool in that like this is what you should look at further. But you're right in that if you really wanted to like understand the biology, you need to kind of look at things over time. In this case, you're right, it's embryogenesis, which is a highly dynamic process. In this case, we're only looking at a single time point. So like day 9.5.
It gives like, it allows for like, what is this building? Kind of double checking things.
Yeah, that's what I told myself to sleep at night as well. Yeah, yeah.
So, concepts like pseudo time and trajectory inference, I mean, there are also like methods to find like a direction.
Yes. Yes, at the end of the day, like as we develop this method, I think the growing consensus in this field is like single cell and spatial transcriptomics are never like the end goal. You never kind of, even though it takes a lot of time and a lot of money, it is only ever like, it has never been used to answer. It's only ever been used to ask questions, right? You always have to have some sort of validation in the end. And yeah, to your point, you kind of put it beautifully, which is like, at the end of the day, we're trying to develop methods that allow you to ask maybe more specific hypotheses in the end. Yeah. As someone who comes from a modeling background, that's kind of the best you can hope for. It's very hard to, I think, answer either using purely like modeling approach, or purely computational approach, or even a purely experimental approach at the end of the day, right? Like, but time is finite and resources are finite. Yeah. But like that is the long-term goal of it eventually, to say something about what you're saying.
Please continue. I mean, it's beautiful work. We're just trying to make it.
No, please, no, I
One
appreciate
more
the
question on
questions.
this. So I see you have 1, 2, 3, 4, 5, 6, 7, 8 genes.
Yep.
In flow. So basically that's one of the ligands in there. And then you create these modules, you highlight four of them. And then you have this sonic hedgehog
Yep.
outflow.
Yep.
And is there anything regarding the directional? Let's say some of these 8 actually increase, or some of these 8 actually decrease the outflow of sonic hedgehog, or is same direction?
Great question. I'm gonna get to that
Okay.
effectively. Yes, I will get to that. Yes. Yes. In short, yes, as I will hopefully show over the next few slides. What was going so yeah, basically, so we by looking at the network and kind of backtrack where like you think the drivers of Sonic Hedgehog might be, and then in converse, you can also look like what is happening downstream. So the idea here is like Sonic Hedgehog seems to be affecting much more downstream than is being regulated by upstream, which kind of makes sense in biology. And yeah, as you can see, like Sonic Hedgehog, its effects are kind of promiscuous in the sense that a lot of different spots actually are receiving Sonic Hedgehog information at this time. And so a lot of different things seem to be depending on receiving Sonic Hedgehog signaling.
One more thing, sorry.
Go for it.
What is the interpretations, like biological interpretations on the intercellular modules? Do you consider those, like say, within the same modules, they are actually the multiple ligands actually works on multiple receptors, or like are they like having this interaction effects, or is that how you consider the interaction?
For the modules?
Yeah.
So the intracellular modules are effectively just like spatially co-expressed genes. And the idea is like those would encapsulate some information we heard at the time about meaningful transcription factors. In this case, the the modules correspond to both different anatomical regions, but also different biological cell states. So they're our way of kind of incorporating cell state information without explicitly defining discrete cell type annotations. So
there is a possibility of those co-expressions and then co-injections, but it's not like truly defined that way.
yes, yeah, basically like, yeah, you would have to go and explore dig deeper
Yeah.
effectively. Yeah. What was the result? Okay, so after we studied these graphs for a little while, kind of, the point is like, yeah, you get these networks, and that's interesting and all, but you would still like to know something a little more specific, which is like, every collaboration I have at the end of the day, I'm supposed to give a list of genes that collaborators can go and test, justifiably so. And so how do you do this using this method? For example, so each of these modules have different genes that kind of comprise these modules. If I look at them, for example, here, you can basically say, because I know that different signals, like the main downstream activated genes are transcription factors, I can look at the transcription factors that are most enriched in these different modules and fit, I stole this approach from Jushen, but basically you fit a random forest model. So the idea is like, of these transcription factors, as you model them as predictors of sonic hedgehog outflow, and then you rank them based on like feature importance. And then you ask like, which are the most predictive of sonic hedgehog outflow? So in this case, these are the sort of top 5 that are most predictive. It goes from 0 to 1. So the ones more on the right are meant to be more predictive of sonic hedgehog outflow. The way we kind of validate this is like the standard way, which is you just Google to see if there's been any sort of implied relation. And luckily, the FOXA transcription factors are very famously involved with sonic hedgehog signaling. Thank God. What was interesting is that it also discovered that there are 2 other transcription factors, MYC, which is relationship to cancer, and Z33H7A. I think it's a zinc finger, or could be wrong, but they haven't been previously reported, but in certain contexts might be relevant to sonic hedgehog. So in this case, the assumption is that when we kind of go into the right, they're also like positive predictors of sonic hedgehog signaling. So they are, I think, as I understand it, they are meant to be like an increase of like activation of FOXA1 and FOXA2 should correspond to an increase in sonic hedgehog expression, for example. We can conversely look at what happens downstream by doing a different thing. So instead of fitting random forest models, we kind of stole this approach straight from pseudotime, which is you fit generalized additive models, you fit splines effectively as a function of the received sonic hedgehog. There are multiple transcription, like many transcription factors. For brevity, I'm showing the ones with high experiment correlation. So the ones at the top are effectively those that seem to be negative correlated with sonic hedgehog signaling flow, and those at the bottom are positively correlated. So the idea is that those at the top may be inhibited by receiving sonic hedgehog signaling, and those at the bottom seem to be enhanced by receiving sonic hedgehog. Yeah, so we can get some quantitative information out of it. These ones I'm pointing out are the ones that have been in the literature, I believe. Some famously so thank God. And we're going to come back to those in a minute, because actually I've kind of dashed over it, but some of these appear to actually be, for example, FOXA2 seems to be both regulated downstream of sonic hedgehog as well as actually upstream of sonic hedgehog. So when you kind of put together, it maybe suggests that actually sonic hedgehog is able to regulate itself via FOXA2. So in this case, it's self-activating, so it actually promotes its own expression via its own cell-cell communication, which I think is known to do. If you go through the Turing patent literature.
So this just means FOXA2 regulates sonic hedgehog.
Yep.
We should then activate FOXA2, or is this downregulated or upregulated?
Upregulated, yeah.
So it's like a looping between these two.
Yeah, it's self-promoting effectively. Yeah.
That's interesting.
It is. Why? Yeah. So kind of going further into that, so that was about self-regulation. We can also look at asking ourselves, well, can we ask a similar question about 2 different signaling pathways? So to kind of go back to it, there are many upstream drivers of sonic hedgehog, 8 here, and there are many downstream targets of sonic hedgehog. If you stare at these graphs long enough, you'll notice that some variables appear in both. BMP4, CXCR12, IGF2, insulin growth factor, midkine, and Wnt5a. And so for context, like I come from the world of Turing patterns and activator inhibitor modules. And so when we're developing this like package, we were asking ourselves like, is it possible to discover Turing patterns or activator inhibitor models from data directly in this way? And so this is what really motivated this question. And to kind of do that, what we looked at, well, first of all, there are different pathways that seem to have a bidirectional relationship with Sonic Hedgehog, 5 in this case. And so the way you would validate whether it actually could happen is by looking at the transcription factors, the ones are expressed in the modules. And so, yeah, to kind of specify better, which is like, of these like ligands I've highlighted, which of them have downstream targets that are also upstream regulators of Sonic Hedgehog? Inversely, which of those have upstream regulators that are also downstream targets of Sonic Hedgehog? So which have regulate certain transcription factors that basically link them to Sonic Hedgehog in the previous slides that we showed. One of the nice candidates, and one I also kind of know from the literature, is Wnt5a. So Wnt5a, Sonic Hedgehog, both important in morphogenesis, limb development, growth, all sorts of stuff. And the idea is like, when you look at these, and you can also work out the correlation, but the idea is like, receiving Sonic Hedgehog seems to be correlated with an outflow of Wnt5a. Just this was like the initial sanity check. They at least seem to be spatially co-expressed. And conversely, if you're receiving Wnt5a, it seems to correlate with expressing Sonic Hedgehog. But we can kind of go further with this. So for example, sorry, go
For
on.
the last slides, is that inflow and outflow, is that expression level or?
Outflow is expression level, inflow is based on cell-cell communication inference. So the idea here is like we can play the same game we did with Sonic Hedgehog, but now we're looking at Wnt. So Wnt5a regulates Sonic Hedgehog through 3 different modules, 2, 5, and 11. The names don't matter, but what does matter is the different transcription factors that appear. So these are the modeled effects of the downstream targets of Wnt5a, and one of the ones that appears is MYC, which I know I've been going through this fast, but was basically regulated to be implied to be an upstream regulator of Sonic Hedgehog. So the idea is that Wnt5a is actually inhibiting this transcription factor that promotes Sonic Hedgehog, if that makes sense. Yeah. So the point is we have a connecting transcription factor that's upstream of Sonic Hedgehog and downstream of Wnt5a. So we have at least one link of Wnt5a going down to Sonic Hedgehog. And in converse, we're going to play the other game, which is like looking downstream of Sonic Hedgehog, which of those seem to predict outflow of Wnt5a? And of those, there's at least 3. FOXA2, NKX, all ones that are known in the context of Sonic Hedgehog as downstream factors. And again, like we find that they're positively correlated with Sonic Hedgehog inflow, or rather Sonic Hedgehog inflow drives expression of these 3 transcription factors. So we now basically what this does is it connects Sonic Hedgehog to Wnt5a by these 3 transcription factors. To put all this together, effectively what you have is what I'm calling an activator inhibitor module. So Sonic Hedgehog basically expresses itself, it diffuses, binds to patch 1, GLI1 in space, and leads to an inflow of received signals in other parts of the tissue. And based on what we like analyze, it can then basically reactivate Sonic Hedgehog via FOXA2. And moreover, receiving this signal via these 3 transcription factors seems to activate an outflow of Wnt5a, which then in turn diffuses, binds, you get an inflow of Wnt5a via cell-cell communication, and receiving that Wnt5a actually inhibits Sonic Hedgehog via MYC. So the point is here we have one self-activating module, the activator, Sonic Hedgehog, and the inhibitor, which is Wnt5a. And effectively what we've done is kind of reconstruct an activate inhibitor module directly from the data. So yeah, this is a personal favorite result of mine, but that's because I come from Turing patterns and French flag models and stuff. Do you guys do Turing patterns in coursework? A little bit.
Got
Okay, cool. Okay, I'm not talking complete nonsense. Good. Okay, so to summarize this hodgepodge talk, like basically we have a method FlowSig that takes gene expression and cell-cell communication inference and spits out a network that describes signal-driven dependencies between intercellular signals and their intracellular mediating modules. And you can use it to basically gives you complex networks that you can then do a bunch of inference on and post processing or downstream analysis, and you can make some interesting biological discoveries from them. So a lot of the extensions so yeah, FlowSig at the moment is an exploratory tool. Ultimately, what you'd really like it to do is make some predictions, particularly some quantitative predictions. That's the ultimate goal. If I turn off Sonic Hedgehog, what happens to BMP? And if I turn it off by like, if I halve it, what is the resulting effect in terms of like the quantitative effect on BMP? But to do that, as Megan pointed out, like you have to account for the inherent timescales and time lags between these different processes. And so currently FlowSig can't do that, but that's like the long-term goal, to really develop a model that can kind of generate these dynamic predictions. And how do I put this? It might be that we basically had to ditch the entire framework. We'll see. But yeah, that's kind of what I would like to do with this in the future. Second relates to what Jutuan was talking about, which is this method has only used transcriptomics data to infer cell-cell communication, but the reality is like there are so many different scales of information that actually drive cell-cell communication. Like, if we're talking about gene regulation, we're really talking about chromatin accessibility. So we would really like a way of incorporating data from ataxic measurements. And also kind of the big elephant in the room of these methods is that actually communication occurs at the protein level. And we would like a way of also incorporating protein and also phosphoprotein data, which is a better measurement of also signal inflow. And also there's metabolites, endocrine signaling, all that good stuff. So yeah, there's a lot to account for either by extending the types of input we can put in and also the framework altogether. I'll end by just thanking people. I realize this is a photo of the lab the most recent photo of Ching's lab that I have, and I realized it is 2 years ago, and more than half of those people have gone on to better places. I hope to join them. But yeah, a lot of smart people have come through this lab and have ended up in very successful places. And also I'm going to thank Omoko, who's a collaborator of ours, and Yuan Chen, YC, her postdoc. They did a lot of I didn't show up, but they generated a lot of the experimental data that was used as validation. They're very nice people to work with. Also at Neuro, if you guys know. And so, yeah, I'll end up by saying, yeah, the paper's out if you want to read it and ask questions. There's a Python package that I've been meaning to make better, but I have other tasks right now. But thank you all for listening, and I'll take any more questions.
That was amazing.
Yeah, I saw notifications. So
There was a comment,
go for it.
but it was like 30 minutes ago.
Oh,
Vincent said, it's like you're learning the divergence of communication of self. But I think
Can
that
you
was
guys
in
hear me?
reference to one of the slides.
Divergence of communication. That's a cool way of putting it. Yeah.
Can you guys hear Zoom people?
uh,
In the volume?
test.
Okay,
Hey,
we can
Vince.
hear.
Oh, sweet. Hey, Axel, sweet work.
Thank you.
Yeah, no, I was just saying the divergence thing was like a cute little mathematical thing. But I got some other questions because this is really cool work. So can you like specify, so you did like BMP4 on the gene level. I guess can you specify families as inputs and say like what are those? Like I don't know. Because there's like crosstalk, I guess. Yeah. So is it like you specify the gene and you kind of like figure out what is the inflow and outflow of that cell with related to that gene? Is that kind of correct?
Yeah, so you make a really good point, which is like if we're talking about BMP signaling, for example, as you know, like it's multiple ligands, there's multiple receptors, and it's a very promiscuous signaling pathway. We made a conscious decision, or I made a conscious decision to focus at the level of genes affecting genes because like as someone who does modeling, talking about just overall BMP signaling as a family of pathways makes more interpretable sense. But if I were to give that to say a collaborator like Max Poikas, he would ask, well, which ligand are you really caring about? Which like interaction do you really specifically care about? So in that case, it was developed with the idea of like, the end of the day, I can kind of give a list of genes to like collaborator. But you're right in that like you could tweak it so that instead of defining individual ligands, you could also talk about like aggregate signaling information. So you could aggregate at the level of pathways and ask like, what is the total amount of received like Wnt signaling, BMP, like all those, and then also
Mhm.
do that in terms of signal outflow as well. You would just basically take the sum or average of them. Yeah.
Yeah, yeah, because I think it'd be cool to say like, because there's like questions about crosstalk. So if you just like clustered
Oh, yeah.
by family and be
Mhm.
like, when does crosstalk happen for given a certain cell type? That'd be cool. Yeah, and then it kind of leads to my next question, which is like, if you are interested in say like cancer, for example, in like a cell, and you have some like differentially expressed genes, I guess you could kind of like, I'm envisioning you could like optimize over, or like just ask the question of which communication channels are overlapping over the differentially expressed genes in cancer. Have you guys thought about something like that?
Uh, yes. So short end of the day, like, yes, it in that way you come. There is another method that asks a very similar question, which is NicheNet or NicheNet, I guess it's pronounced in America. Which is like if you have a certain like group of genes that differential express, what were the upstream ligands that like may have regulated the differential expression? You could do the same in FlowSig by just specifying that those differential expressed genes as signal outflow nodes. So that in that way you could ask like, does receiving a certain signal cause a change in the expression of those genes you care about? I should say also like prior to inference, there is preliminary like differential expression analysis to kind of work out like which signal inflows and which signal outflows actually change if you're looking at from like health to cancer, for example.
Mm,
Short answer is yes, you could, very straightforwardly. Cancer is a little tricky though because it with non-spatial data you're talking about normal versus perturbed, but it's like perturbed within reason, and cancer is like, yeah, I still debate how do you do you infer cancer as like a perturbation or something else altogether? Yeah, I guess
yeah,
it depends.
yeah. All right, yeah, thanks for those answers and great work.
Yes. Think we're done.
I had a question.
Go for it.
So I also do spatial transcriptomic stuff, but it's like imaging based, single cell spatial transcriptomics. I was wondering if you could talk a little bit about why that might or might not work as well for this specific tool. Yeah.
I would fucking love if it worked for stuff like Merfish and Seekfish. It's not that it doesn't work. It's that, so for example, with Merfish, I guess that's what you guys are using.
We're using Cosmics, but like
Oh yeah,
same idea.
okay, yeah. Or like even so, I guess it's kind of gone away with Xenium now. But basically, the point when we were developing this is like fish-based methods have much lower throughput. So like you're several hundreds of genes, and usually those genes are based on what you specify via probe set, right? Because of that, most like fish-based data sets don't include information about both the ligand and the receptor. So it's very hard to infer cell-cell communication from those data sets. It's not that you can't do it, it's just that they don't, you're prioritizing different like probe choices. You're usually looking for cell type distributions in space. So you'll prioritize cell type markers rather than say cell-cell communication.
Okay.
Yeah.
So you're saying if the ligand and receptor genes are in the data set, then
Yes,
you would still be able to use it.
absolutely. So I
Got
think with Xenium, I guess that is like 10x is answer the stereoseq. You should be able to do it theoretically. Yeah. Um, like the end goal is something fish-based, to be honest, because of the sensitivity.
Mhm.
It's a matter of time
Yeah.
before the throughput gets scaled.
Yeah, Cosmics currently has like a 6,000 plex panel for human, and then they're trying to come out with 6,000 plex for a mouse, but right now it's like 1,000. It's still not bad, but
Yeah.
yeah.
I had for a while I've been looking at like codex data of PhenoCycle, like the Acquire Biosciences data. But again, I think that's like 50 something proteins. But like that would be the end goal, something like that.
Cool. Oh yeah, let's stop.